I'm Seth Pollack from Northwestern University, and I'm so grateful to be here. I love Selecta. We've done a lot of TCR sequencing in my lab, and I can tell you that we've done a lot of work. Just whenever we use a new technique, we want to see in our own hands that this is reproducible, and we've done all of that, and we think the technology is super reliable. It's affordable compared to some of the other TCR platforms that are out there, and they give you a lot more information too. So we love it. So my lab, we study sarcomas. I'm a sarcoma physician scientist, a disease expert. Sarcomas are rare cancers. In aggregate, they're not that rare. They're about 1% of all cancers, which is as common as some cancers that you don't think of being that rare, like myeloma, which we're hearing about today, probably a similar total incidence, but sarcoma is actually a group of diseases. So some of them really are truly ultra rare, and we usually divide them up into soft tissue sarcomas and bone sarcomas, not for any biologic reason, more for surgical reasons. And in terms of bone sarcomas, there's osteosarcoma and Ewing sarcoma that you might have heard of, and then the other relatively common bone sarcoma is chondrosarcoma, which affects about 300 people a year. It has this, it's actually a cancer of the chondrocytes that make cartilage. This is what it looks like under the microscope. You can see there's these cells that produce this cartilaginous matrix around them. Second most common bone tumor in adults, usually in the axial skeleton. And importantly, there's really no treatments for chondrosarcoma. It does not respond to chemotherapy. There's no FDA-approved treatments. Now, it has responded in the past to checkpoint inhibitors, rarely. So there's a case report that we wrote on a patient who had a great response to PD-1 blockade. There's a case series of bone sarcomas that were treated with a PD-1 inhibitor, and that included a handful of chondrosarcomas, and they had 2 responses in the chondrosarcomas. So 2 responses in the chondrosarcoma, which was actually like 15-20% of the patients, which is not good by melanoma standards or lung cancer standards, but still was enough to make us think that, wow, there's probably this subsection of chondrosarcomas that respond to checkpoint inhibitors, and we've really never seen an inflammatory infiltrate in chondrosarcoma tumors. So we wanted to look more deeply into whether there is inflammation and maybe a subset. And this was part of a bigger project where we're growing out T cells from a lot of different sarcomas, including chondrosarcomas. So we decided to take a closer look at the microenvironment. And interestingly, first thing we did is we looked at some nanostring of just chondrosarcoma samples that we had, and we saw that and you can see the chondrosarcomas in blue over there. It actually, when we looked at all these inflammatory genes, it had much higher levels than some of the other sarcoma types that we looked at. And we were like, oh, that's really strange, because these tumors seem very uninflammatory to us. And then you could see here by this multiplex imaging, the green is the tumor marker, and the purple is the T cell marker, and the yellow is the macrophage marker. That basically, what was happening was that we were doing sequencing of chondrosarcoma tumors, but we were basically including the peritumor area, which was really thick with T cells and thick with macrophages, but none of that was in the tumor at all. And we looked at this by IHC as well, and you can see here, this is like kind of our quantitative IHC technique. And when we did that, we found that, we compared the tumor to the peritumor area, and pretty much all of the T cells were in the peritumor area, and there was a big drop off. There's no statistics shown in here, but I can tell you it was statistically significant every single time. that there was more cellular infiltrates in the peritumor area. And we saw this. We did some geomics as well. But what we wanted to do is we wanted to do TCR sequencing in the tumor and in the peritumor area. And for this, we used Selecta's error assay to do the TCR profiling. And we looked at both the tumor, we looked at the peritumor area, and then we found specific immune clusters that we sampled. And we did basically punch biopsies of the FFPE to do this. And you could see here that we saw a lot of TCR sequences in the peritumor area, which is blue on here. We saw even more in the immune clusters, which were centers in the peritumor area that seemed to be more dense with immune cells. And we saw very little of these TCR sequences in the tumor itself. But then we started looking more closely at this, and what really shocked us was that we found overlap in some of these TCR sequences. So I'm going to show you in just a second, but the TCR repertoire, you've got about they say there's 10 to the 12 different combinations, and there's so many different TCRs that no two people should have any overlap. There actually is some overlap between individuals, probably 10 to 20,000 TCRs that are shared, but in a small sample, you really shouldn't have any TCR overlap. And what we just saw is pretty much every sample we looked at shared a lot of TCRs with the samples from other patients, which didn't make any sense. And some of them were in multiple cases. You can see this one had 8 cases, and these were high frequencies. The title of this slide gives it away. These turn out to be mate T cells, but you could see that it was not a small fraction of the T cells that were that had the overlap. Here's just a slide about public T cell receptors. Public T cell receptors is what we call it when two people share the same T cell receptor. It's something that it will happen if you look at somebody's whole blood and you look all the way down to the rare T cell receptors. There's a number of viral public T cell receptors that have been described to EBV, to CMV, to COVID, and in the presence of an acute infection, those can become more common. But if you just look at healthy individuals, you don't find public TCRs among any of the, any of the common T cell receptor sequences. They're really only the ultra rare T cell receptor sequences that are like less than 10 to the minus 5 of the blood. But we looked at these, and basically we were just so shocked that we saw all this overlap that we started Googling them, and we found that some of these T cell receptor sequences had been described as associated with mate T cells. So mate T cells are the mucosal associated invariant T cells. These are a group of evolutionarily conserved innate-like T cells. They're primarily localized to mucosal surfaces like the GI tract. They can be found in the lung. They can be found in the liver and in the intestine. And they have a restricted T cell receptor repertoire with a lot of the times they share an alpha chain. And that's one reason why it's so nice to use Selecta, because Selecta gives you both alpha and beta chain readouts. And they have a more limited beta receptor repertoire, and they specialize in seeing metabolites that are not like foreign antigens, like regular T cells see. And they don't see their antigen in the context of HLA class 1 or class 2. They see it presented by a protein called MR1, which is a non-canonical antigen presenting molecule. And there's actually an online tool that you can put your T cell receptor into, and it will tell you if it's probably a mate TCR. And when we looked at all of our shared antigens, we found that almost all of them, when we put them into the online tool, turned out to be mate TCRs. And actually, when we looked at all the TCRs we had looked at, pretty much between 10 to 40% in these chondrosarcoma tumors and the peritumor area were actually mate cells based on the TCR repertoire from Selecta Technologies. And then we looked at we went back to our punch biopsies of the tumor and the peritumor area, and we found that the mate cells are present both in the tumor and in the peritumor area. And while there's less mate cells total, because there's just less lymphocytes in the tumor, a higher percentage of those lymphocytes that are in the tumor seem to be mate cells. No, this is still this is TCR sequencing. You see, yeah. We'll see. This is just talking about MR1, and talking about MR1 and the antigens presented by it. And then we didn't do IHC for the mate cells themselves because, but what we did is we looked at, we used the geomics assay that we had and looked for gene expression for the genes related to mate cells. And again, we found that the T cells in the tumor express the genes that we would expect to see with mate cells. Again, within the tumor, with other lymphocytes, it was pretty much only in the peritumor area, but with geomics, we saw them also in the tumor area. And then we looked with MR1, which is for what you look at for the antigen that, for the molecule that presents the antigen to the mate cells, and we found that basically every chondrosarcoma cell strongly expresses MR1, and we also saw this in the peritumor area to the same degree. And then we looked, we quantified the MR1 IHC and found that it was in every tumor except there was very low levels in one of them. And there was no difference in the MR1 between the tumor and the peritumor area. So using Selecta, we found that we can classify chondrosarcoma as an immune-excluded tumor, meaning that there's very few T cells in the tumor itself, but there's actually a lot of T cells that are directly around the chondrosarcoma tumors. And then the AIR assay helped us figure out that these T cells that were in and around the chondrosarcoma tumors were mate T cells, and then we also found that chondrosarcomas almost always express MR1. So we're going to be doing a larger analysis with chondrosarcoma and to try and figure out what these mate cells are recognizing and how to get them to recognize and kill the chondrosarcoma tumors. And then we're going to be doing a large investigation with other sarcoma tumors, again using Selecta's AIR. So this is my lab, and I'm so grateful for all of you for being here. Thank you.